Hello everyone, we are the team number 2 and today we're going to present to you the dimension number 3 analysis activity stage number 1 and my name is Sofia Tirado.
My name is Ángela Acevedo.
My name is Emiliano Ortega.
My name is Mauricio Hernandez.
Okay, so I will start with the cell. The cell is the basic structural and functional unit of all living things. So the cell is the basic unit of life, and well, like it says here, all living things are made up of this. So some characteristics are cell are complex and organized. They have many organs or organelles and different membranes. Then the cell possesses the ability to replicate. This means they can reproduce themselves with another, or simply doing mitosis. Cells obtain energy. This means they can feed up. This depends what kind of cell they are. And cells interact with the environment and also reacts with the stimuli. There are different types of cells. They are the eukaryotic and prokaryotic. The eukaryotic are the animal and the plant cells, which are going to see in a moment, and then are the prokaryotic, which are bacteria and archaea. The animal and plant cell organelles, uh, like I said earlier, these animal and plant cells, they have differences between them. They are from the same type, but they are different organisms. For example, the the animal cell is the most common one, which are considered the bricks in the genetic building, and the main parts are the nucleus, the plasma membrane, the cytoplasm, the endoplasmic reticulum, the Golgi apparatus, the centrosome, the cilia and flagella, and the mitochondria and cytoskeleton. So the animal cell has almost all they share with the plant cells. For example, a typical plant cell consists of a nucleus, a cytoplasm, and very internal compartments they share with the animal cell. And in addition, these cells are surrounded by a plasma membrane that contains ribosome that produce proteins. They have a wall cell, and because this helps them be, to build a more simply a more easy organism, and they have a little membrane that is chloroplasm that generates energy.
The characteristics of living things. The characteristics can be divided into 8 points. One is they are made of one or more cells. The two is they have a metabolism. The third is maintain homeostasis. The fourth is respond to stimuli. The five process DNA as genetic material. Um, the six is they grow. The seven is they reproduce. And eight is they evolve. All of these characteristics needs I think to can be really living thing. But what is the difference between the prokaryote and eukaryotic cells? Well, prokaryotes are exclusive unicellular, which size range from 0.2 to 2 in diameter. They are usually process a chemical complex cell wall and have a nuclear region instead of a nucleus. Their ribosomes are smaller and spherical, and their DNA is circular. Prokaryotic cells lack of mitochondria, endoplasmic reduction, and organelles like lysosomes and centromere. They reproduce via binary fission and may have a small phalangia. Plasmids are commonly found in prokaryotes. In contrast, eukaryotes can be either unicellular or multicellular. If a cell wall is present, it tends to be simple. Eukaryotic cells can obtain a true nucleus housing, linear tinea. Their ribosomes are larger and linear, and they contain mitochondria, endoplasmic reticulums, and various organelles, including lysosomes and centrosomes. Eukaryotes can reproduce both asexually and asexually through mitosis typical processing layer. Plasmids are uncommon in eukaryotics.
Now I am going to talk of the history of microscope. In 14th century, spectacles first made in Italy. In 1590, two Dutch spectral makers, Father Anson Tim, Hans and Sir Jason, created the first microscope. 1677. Robert Hooke's famous Micrographia is published, which outlined Hooke's various studies using the microscope. 1675. Enter Anton van Leeuwenhoek, who used a microscope with one lens to observe insects and other specimens. Leeuwenhoek was the first to observe bacteria. 18th century, as technically improved microscopes became more popular among scientists. Part of this was due to the discovery that combining two types of glass reduced the chromatic effect. In 1830, Joseph Jacobs discovered that using weak lenses together at various distances provided clear magnification. In 1878, a mathematical theory linking resolution to light wavelength is invented by Ernst Abbe. In 1903, Richard Sixmondi invents the ultramicroscope, which allows for observe specimens below the wavelength of light. In 1932, transparent biological materials for the first time using Fritz Chernyk's invention of the phase conference microscope. In 1938, just 6 years after the invention of the phase contrast microscope, comes the electron microscope, developed by Ernst Rutherford, who realized that using electrons in microscopy enhanced resolution. In 1981, 3D specimen image possible with the invention of the scanning tunnel microscope by Gerard Bing and Heinrich Rohr. The cell theory. Cell theory is a scientific theory that states that all living organisms are made of cells and that all cells come from other cells. What does the cell theory say? Cells are the basic unit of life. All living things are made of cells. All cells come from other cells. Cells are separated from their surrounding by a cell membrane. Cells contain genetic material that is passed on to other cells during cell division. All cells are similar on their chemical composition. The activities of an organism are the result of the combined action of an individual cell.
Okay, now I'm going to explain the different forms of cell transport that exist. Cells need to move substance in and out to survive, and there are two main types of cells. Passive transport moves molecules from high to low concentration, which are 1. Diffusion molecules. These molecules move naturally, like oxygen entering cells. 2. Facilitated diffusion molecules. These are the larger molecules and use proteins to pass through, such as glucose. And finally, 3. Osmosis molecules. In this, water moves to balance concentration. Now, we are going to talk about the second type of cells transport that exists, active transport, which moves molecules against the concentration gradient using energy ATP. This transport has different molecules, which are 1. Active transport molecules. In these, proteins pump molecules, such as the sodium-potassium pump. 2. Endocytosis molecules. In these, the cell wraps substance to put them in, like white blood cells trapping bacteria. And lastly, 3. The exocytosis molecules. In these, the cell releases substance like hormones living in a cell.
For levels of organization, we have that all body functions rely on a coordinating system performing different tasks simultaneously. With this, we start from subatomic levels with the atom. A set of atoms intertwine, form a molecule. The molecules in turn will form organelles that will be integrated to build the cell parts. A lot of cells with similar functions is known as a tissue. that are the building blocks or organs which are the structures that make the systems, that are the digestive, the respiratory, the urinary, the skeletal, muscular, etc. And we have the homeostasis. That is the characteristic of a living being and non-living being, is the ability to maintain their internal conditions despite the condition of the external environment. Basically is the adaptability of a body to its external conditions.